Gonna finish up some loose ends here in reproductive, spermiogenesis, meiosis, fertilization, and kind of wrap things up. So meiosis for both the gametes, both our oocytes and our spermatocytes, go through meiosis. So we're going to go from a situation where we have a 2N cell, and we're going to duplicate that cell through our meiosis 1. We're going to take those 2N cells, divide them up, and then we're going to end up with 4 1N cells. So we're going to have half of the genetic material, 23 chromosomes, in all of our cells at that point. Again, in the sperm, all 4 of those cells are going to be functional cells. In the oocyte, there's only going to be a single cell that is a viable oocyte, and the other 3 are going to be polar bodies. So for spermatogenesis, we already looked at oogenesis with the female. Spermatogenesis goes along, and again, said before that we kind of start out with something that kind of looks like a cuboidal type of a cell that is there. That cell is going to then duplicate as we go through our process here. Our primary spermatocyte, kind of like our primary oocyte, is a 2N cell. That cell then goes through our cell division one more time, and we're going to generate 2 secondary spermatocytes that are now 1N. That then has our final cell here, where we have now a set of 4 1N cells in the end. And those are then going to go on to have the potential for fertilization that's there. Through this process of cell division, the cells themselves are going to end up having essentially just a head. They will have a mid piece, and then they'll have a tail. And so the head contains all of the chromosomes that is going to be needed for creating that final cell after fertilization. In addition, the head has kind of like a cap on it. And so this is where we have what we call the acrosome. And this acrosome has enzymes in it that's going to allow it to actually penetrate into the egg. The egg has the zona pellucida, the corona radiata on it, and it has to be able to get through those, as well as that of the plasma membrane of the oocyte, in order to actually fertilize the egg. The mid piece is all about essentially the engine. So this is mitochondria that are there at the very end, kind of going into the tail. We then have centrioles there, and those centrioles are then going to be able to make the sperm mobile. That's there. So throughout this process, as we start to get down to our spermatocyte itself, that's there. Some of this excess cytoplasm is going to be lost on its way down towards the epididymis, where it's then stored as a non-motile cell here in the last cell here, where we have, again, the head, the mid piece, and then the actual flagellum itself on the cell there. For fertilization to occur, we saw that ovulation occurs at day 14 of the female cycle. And at that day 14, the oocyte is then ovulated. It's going to start its journey down the uterine tubes, awaiting for it to be fertilized by sperm. And here we have kind of a time game, in a sense, where timing needs to be just right for fertilization to occur. The oocyte's going to typically live somewhere in the neighborhood of 24 hours. So it's going to remain viable for that 24 hours or so. And the sperm is going to be viable anywhere upwards of 72 hours. So we've got kind of a 3-day window, in a sense, where fertilization can kind of occur on there. In terms of the sperm, we've got basically a 1-day window in terms of the oocyte. And granted, our bodies don't read the book, so they're not necessarily always going to be spot on in terms of those numbers. There's all kinds of cases where fertilization has occurred, where couples definitely know that they did not have any kind of intercourse or anything within that time frame, and yet they still became pregnant. So doesn't always exactly line up in terms of the timing there, but these are the generalizations for those cells under kind of usual conditions that are there. If the sperm does find an egg, so the sperm has kind of a 50-50 chance to begin with. It has to pick the right uterine tube on its way up through the uterus, and if it picks the right uterine tube, then of those 120 million plus cells, there's the potential that about a million or so actually make it to the oocyte, and fertilization can potentially occur. We then have that zygote. That zygote then can start to make its now roughly 6-day journey or so, continuing on, by this point, maybe 5 days, down into the uterus. Once sperm is ejaculated, it's not a guarantee, like I just kind of said with the 50-50 there, that it's ever going to make it to the sperm cell or to the oocyte. So the sperm, if it doesn't coagulate well, it could immediately exit the vagina. There could be environmental factors, so there could be bacteria, there could be a overly acidic environment that's there. The cervix itself may not be open enough, and so it may not actually get into the cervix there. It might get to the uterine tubes, but it again picks the wrong one. And so if ovulation has occurred in, say, the left side, and the sperm make their way towards the right side, they're going to find nothing there to fertilize that's there. Once the sperm actually does find the oocyte, then we have the potential for fertilization. And then it's going to come down and implant into the uterus there after that fertilization has occurred. But for fertilization to actually happen, again, like we said, the head has to be able to get into the oocyte. And for that to occur, we have to have a reaction between the sperm head and that of the oocyte itself. And so the oocyte has the corona radiata, it has the zona pellucida surrounding the cell itself that's there. And the sperm cells basically need to be able to get through all of those layers. And so it's going to bind to the zona pellucida, at which time it's going to start to release its enzymes from the acrosome that was on the top of the head. That's there. And that's going to basically dissolve that zona pellucida. That's going to allow it to start to come into contact with the oocyte. On the surface of the oocyte, there are receptors. So just like we've talked about in other types of tissues, whether it be for hormones or neurons or chemicals, we have these receptors on the surface of the cell. Those receptors help to bind the sperm cell to it, and then the sperm can start to get in through into the actual oocyte's plasma membrane itself. And so alpha proteins cause it to insert into the membrane. It's now, in a sense, kind of trying to get through the membrane itself. And once it does so, it then can deposit its chromosomes through its cytoplasm into the cell itself. Once that one sperm makes it in, so here we have a oocyte with many, many sperm surrounding it. Once that has occurred, the oocyte doesn't want to have more than one sperm. In other words, it would have now 3 sets of chromosomes to get inside. So in order to stop that, the oocyte's going to go through a couple of different things. The first is going to be what we call the fast block. The fast block is essentially going to be a depolarization of the oocyte. And so this is now, as it depolarizes, it makes it so that it's no longer attracted, or the sperm cells are no longer electrically attracted to the oocyte. And so they now have the same charge, so they're going to repel each other. The slow block is where all of those receptors on the outside of the oocyte become destroyed. And so it's going to break down all of those receptors so that another sperm cell doesn't have anything to bind to. If it doesn't have anything to bind to, it can't get into the cell. Once the chromosomes from the sperm are now inside the oocyte can complete meiosis II. And so now it's going to continue on through meiosis II. Once it does so, we get 2 cells. We get the last polar body, and then we get our zygote. And so the second polar body, the first polar body we got after the first cell division, the second polar body we get at this point, and that's essentially going to be the last cell that is created from our original developing oocyte itself. That's there. The two pronuclei from the oocyte and the sperm are eventually going to come together. So we have the pronuclei that are there, and they're going to come together and fuse, and we now have that one final cell at that point inside there. Fertilization has occurred. Cell development continues from here. And so we get two daughter cells. Those two daughter cells are then going to divide again. By the time we get up to 16 cells, we have what we call a morula. And so this is now still traveling down through the fallopian tubes. We're roughly 3 days in at this point. And at that point, we have a number of cells that are now dividing. And now we get up to 100 cells. The 100 cells is what we call a blastocyst. And so by that fourth or fifth day, we're now entering into the uterus, and that blastocyst is now going to have the ability to bind with the uterus. And so we get implantation. The blastocyst is a big sphere of cells. So we've got a whole bunch of cells along the outside. And this is going to start to differentiate into two sets of cells. So at this point, we have two components to the cells. One is the trophoblast. This is the outer layer of cells. So we've got the sphere itself. And then on the inside, we've got the inner mass of cells that are there. The trophoblast is what's going to bind to the uterine wall. So this is what's going to start to become the placenta. And then the inner mass of cells, that's what's going to start to become the embryo. So moving on down through the uterine tubes towards the uterus, fertilization occurs at one point somewhere along the way, and then we start the clock in terms of post-fertilization occurring. So we have our zygote there, 4 cells, 8 cells, 16 cells, we have our morula, 100 cells, we have our blastocyst. The blastocyst comes down and we have our inner cells, cell mass, and we have our trophoblast on the outside. Trophoblast eventually binds to the uterus there, and we get implantation occurring inside the uterus. Just a chart to kind of look at in terms of the terms of the names of the different timings for our post-fertilization cells, and then eventually embryo and fetus, and then after birth and so on. In terms of male versus female, we've talked about this in terms of separating the two out. Differentiation between males and females, we talked about homologous types of tissues that we find between males and females that are there. When it comes to differentiation, the component that is going to determine whether we are male or female is going to be the presence of testosterone. And so essentially, if we take even an XY zygote and inhibit its ability to produce testosterone, we're going to end up with a sexually differentiated female. That's there. And so the presence of testosterone gets us to the male point. Essentially, the default is the female there. Through that differentiation point, and in terms of that, the development of the reproductive organs themselves, it does take quite some time for the overall differentiation to kind of finish. And so sexual differentiation itself goes along. And here we can see by the 10th week, essentially externally, the genitalia still relatively look the same. The only difference is that essentially kind of the size of the external genitalia hasn't changed. You can see the folds there of the what will become the labia majora or the scrotum, what will become the ventral penis or the labia minora, and so on. So all the structures are relatively the same. Once we get closer to birth, and so we get further on out, 7 months and on or so, we now start to see true differentiation in terms of the external genitalia. that are there. You can see early on as well that whether male or female, essentially the ducts are both there for both male and female. And as they start to differentiate, they're either going to become degenerated or they're going to be functional, depending upon the types that we have there for either the vas deferens, the ductus deferens there, or becoming things like the round ligament and so on. And so the differentiation there goes inside as well. We talked about the difference between or where fertilization occurs inside the fallopian tubes, and then implantation we said occurs inside the uterus. But sometimes implantation doesn't always happen inside the uterus itself, and so we get extra uterine implantation, and we call this an ectopic pregnancy. Essentially, the ectopic pregnancy can occur just about anywhere that's not the uterus, and even sometimes in the uterus, but not along the wall of the body itself either. And so it could be in the fallopian tube because there's no true connection in a sense between the fallopian tube and the ovary. We could also have abdominal pelvic implantation occurring that is there. And with ectopic pregnancies, typically these are not viable. They're going to cause some sort of damage to either the woman, or they're going to cause a lack of blood supply to the developing embryo fetus along the way, depending upon how far along it gets. So usually these are terminated. Once in a while, though, something like an abdominal pelvic ectopic pregnancy may go full term if it finds a good blood supply inside the abdomen or the pelvis that's there, but usually not. And with that, we end our look at the reproductive system.